What we do is we provide back to Immunobiotech a certificate of testing of what we do. This is the information they give us, which we put into it, and then these are our tests with our specifications that we've drawn up. I must say that we drew these up as passes in discussion, and also using our knowledge as what we've got. And these are real life results of one of the batches that we sent back. And we also seek out C surveys on some of the material involved to make sure that there's no hepatitis B, hepatitis C, or anything like that. So we're fairly confident and also quite rigorous in our approach to the testing that we do on the map. To give you an example, and if you can just bear this in mind here, this is the start, this is the finish, and what I'll do is I'll try and show you one of the time lapse that goes on. So here's the lump. I don't know if that can be seen. It would be handy if the lights were down, but that's the lump that we saw in the original. The small cells here which are moving are the activated macrophages. That's it. Thank you. And you can see the attack in it. And if you can focus on a little bit of the monolayer, as the movie goes through, you'll just see what happens to it as it becomes distended and the cells lose their sort of connectivity, if you like.
Rod, I think we ought to point out that the monolayer is a cancer monolayer.
It is. It's an MCF7 breast carcinoma cell line. So it goes out of focus because I get a little bit of condensation on the roof of the 12-well plate.
Could you use the microphone, please?
Certainly.
Thank you.
So,
yes.
So the breast carcinoma cell line is there. You can see that on the basis. The round cells which are buzzing about are the activated macrophages. They've been activated with 100 nanograms per mil for 72 hours before application. And then this is a time-lapse photography with a frame collected every 2 minutes. And you can see the effect as it goes on. So we see this on almost every batch that we've got. This is just one of the examples of the batches that we do. So my quest now is, well, Emma and I at Macro are trying to learn about macrophages and how they interact, the cell signaling that goes on and what they actually function and how they do that. This is an expectation of what we intend to see, and if we don't see this, then it doesn't pass, basically. So that's one of the sort of testings that we do to make sure. So you can see the monolayer is almost practically cleared into a big syncytial mass. Obviously, the speed of the degradation of the monolayer can be affected by concentration of the cells of one or the other type, and also how much Gcmaf we actually activate with. It's fairly comprehensive destruction of a breast carcinoma cell line, really. When we get our other carcinoma cell lines going, we'll do the same again with our controls. What we have done is we've done activated urine macrophages on normal breast cell lines, and you don't see anything like this at all. And we've done that visually at the minute, observation-wise, in the microscope, but we're intending to do the same kind of time-lapse photography for that, so that we can include that as a control of non-activated, normal breast cells against activated macrophages. Because one of the issues might be that, oh well, if you activate macrophages, they'll go around affecting every cell line, but that's not what we've seen at the minute. They're fairly busy little creatures, really, the macrophages. And when you talk about big eaters, the cancer cell lines are quite large in comparison, so they're not worried about attacking them at all. Yeah.
It's been on the website for 6 months now. Yes.
I think some of it's been on there, yeah. So it's pretty conclusive in our eyes that breast carcinoma cell line is destroyed, eaten, removed in a matter of hours with activated macrophages using Gcmaf.
Yeah, excuse me. Have you tried to use any of this data to get like FDA approval so it can?
No, not at the minute, no.
Why?
Why? We're not that robust at the minute, I don't think, and I think we're not that robust in terms of what we know, we've only got the one cell line and also murine macrophages, so we really need to do it with the human macrophages, which I don't think will have any issues at all, and a number of other carcinoma cell lines.
The next dose of macrophage is still in the same picture and on the same, the same media, the same sample.
No, we've not done that. We as in a tissue culture lab, you make your media as you go along. So we have the same batch, if you like, of basal media. Sometimes the serum changes. This is in serum-containing media, and at the minute, no, we don't monitor vitamin D that's in the media, which we're intending to do as part of the control work. But we have a number of controls in terms of reducing serum, going into serum-free and that sort of thing to make sure that we can directly compare, which is part of the issues of trying to get any kind of acceptance.
But it would be interesting to see the same and when it starts.
I agree, yeah. This is, just to give you an idea now, this we spoke about, this is breast carcinoma, MCF7s, and this is where we added Gcmaf alone, and then after 72 hours you can see that happen. So there's no macrophages in this at all, and a small, shorter video.
Is it similar concentration or loss of concentration?
A similar concentration of activating macrophages, yeah. You have to choose somewhere to start. I think Marco Rigaria's group were talking about 40 nanograms per mil. We decided on 100 to work down from there.
About the concentration, did you do any tests about the concentration and the tests and the dose at least?
In terms of Gcmaf?
Mm-hmm.
Well, the vials come in at a certain concentration and we take that as read and use it in that fashion, dilute it straight down. It's so the buffer that's used there doesn't
But there are
affect.
other papers who read, your company speaks about 100 micrograms for one dose. The other companies say you need 10 times this much.
Well, the work that we've done, we can get an effect at 100 nanograms per mil, 40 nanograms per mil.
Nanogram.
For the direct effect, this is 100 nanograms per mil, but 40 nanograms per mil does the
In
same.
100 nanograms per mil in
Final.
In this vitro test or in the body?
In the actual 12 well culture.
100 nanograms.
Yeah.
But it's a very high concentration compared with the whole body, for example.
Yes, it is.
Which you recommend also 100 milligrams.
Yeah, I've
which
not
is
done
10 grams and my whole body is 70 grams,
Well,
70 kilograms.
I understand what you're saying, but I've not done a dose depression or a descaling process to find out where there's a cut-off point. I've done a few trials on producing curves, and there definitely is a point of where suddenly the activity stops. But that's in preliminary data at the minute, and it's only again on the one carcinoma cell line.
Okay.
In conjunction with the group, you might see some of the posters around that are being produced, and just to highlight what David said, we've had a number of posters and papers accepted. I think there's 8 in all. There's a list there. It shows the collaboration, and we're sort of forging with Marco Rigaria's group in the University of France, and we're very pleased to be associated with that. We've also, in discussions with the University of Sharjah in Dubai, we're starting to enter into potential collaborations there, so we're starting to spread our network, and hopefully we'll gather more information and be able to do more in that respect. So I just wanted to highlight to you the tests that we do, some of the results that we got for consistency and safety, and then for us to be in a privileged position that we see this week in, week out in terms of the clearing of carcinoma cells with Gcmaf.